Assalamualaikum and a very good morning. My name is Muhammad Arif Aziz bin Muhammad Shahid. I am a student of Bachelor Honour in Biology at UiTM Shah Alam. Today I am going to present my final year project on the ecological effects of ammonia on lichen and algal community. I am under the supervision of Association Professor Dr. Asmina Ismail. This study focuses on understanding how ammonia affects the ecological balance of lichens and algal communities. These organisms are crucial indicators of environmental health and play a significant role in their ecosystems. The problem statement of this study is that the amount of ammonia produced by agriculture and industrial operations has steadily increased, posing a significant threat to the terrestrial ecosystems. Moving on to the objective of this study. There are three objectives. First is to assess the effect of ammonia on species composition of lichen and algal communities in urban and rural area. Next, to examine the pH level change on the composition, species coverage, and density of lichen and algal communities. Lastly, to determine the relationship between bark pH and the concentration of ammonia towards lichen and algal communities in urban and rural area. On to the methodology. The site that has been selected was Sekolah Kebangsaan Titi Dayajaya, Shah Alam as the urban site, and Pusat Kaji Cuaca Batu Embun Jerantut as the rural site. Both sites contain a CAQM station, which is a short for a Commission for Air Quality Management, which air quality data are collected in real time. And for sample collection, it consists of 10 trees per site. And for the species identification, the stereoscopic and light microscope were used. The stereoscopic microscope were used to identify the lichen, and light microscope were used for algae identification. Next part of the methodology are the parameter calculation, which there are two equipments that had been used. For the first one are Aerocoil Series 200 to monitor the ammonia level on both sides, and pH meter were used to collect the level of pH of the bark. Then the statistical analysis, which are t-test, lichen frequency count, coverage, density, and abundance, and also algal density. In this discovery, there were 10 species that were found in an urban site, which as depicted here. There are Diurnaria aplanata, Diurnaria picta, Graphis liberata, Graphis scripta, Lepra incana, Lepra labificans, Leperculon microscopicum, Paranula paranuloides, Altonia albovirescens, and Pammoterma tinctorum. And the species of lichens in rural sites are Diurnaria aplanata, Diurnaria picta, Graphis scripta, Graphis intricans, Lepra incana, Lepra labificans, Pammoterma tinctorum, Altonia albovirescens, Licidia lithophila, Protoblasteni rupestris, and lastly Chrysotrix candelaris. There are three types of lichen: crustose, foliose, and fruticose. But only two are present in the study, which is crustose and foliose. There are more crustose type lichen than foliose, and the number of lichen found in urban area are higher than rural. This is because in urban areas, crustose lichens are often more prevalent compared to rural areas. Next, figure 2, species richness in urban and rural site. There are no significant difference between them. Same goes to the other three parameters. In figure 6, it shows that species richness between sites by species. It is stated that the highest species that were analyzed were Althonia albovirescens and Graphis scripta. Althonia albovirescens is highly adaptable to a range of environmental conditions, including those with higher levels of ammonia. Same goes to Graphis scripta. In figure 7, species coverage between sites by species were analysed. The lowest were Dineraria pincta at 1.67% and Chrysotrix candelaris at 1.04%. Dineraria picta shows a very low coverage at only 1.67%. This lichen is particularly sensitive to environmental changes and pollutants, including ammonia. In figure 8, the species abundance between different sites, specifically looking at the number of individuals of each species present in urban and rural areas. Lepraria incana is the most abundant species in urban areas with 15 individuals. This species is well adapted to environments which often have higher levels of pollutants such as ammonia. Graphis scripta shows the highest abundance in rural areas with 41 individuals. This species thrives in less polluted, more stable environments, typically found in rural settings. Figure 9 shows species density between sites by species. Highest recorded of species density in urban site is Lepraria incana with 248.33, and highest in rural site is Graphis scripta at 676.67. Next, on to figure 10, the table stated lichen frequency of urban site. The highest frequency are Arthonia albovirescens and the lowest are Dineraria picta. There are differences in frequency of lichen species, which will shown in this table. The species Chrysotrix candelaris, Graphis intricans, Licidia lithophila, and Graphis intricans are present only in rural site. On to the abiotic parameters, which are bark pH against sites and ammonia against sites. In the bark pH against sites graph, the standard error mean indicates that there is a significant between urban and rural site. However, the graph ammonia against sites shows no significance. This shows the correlation between ammonia and bark pH in urban and rural sites. In urban sites, the higher the bark pH, the lower the ammonia level. However, in rural area, the higher the bark pH, the higher the ammonia level. These are the algae genus in urban site. Desmococcus species, Chlorococcum, Trentepolia, Treboxia, Apatococcus, and these are the algal genus in rural site. Desmococcus, Chlorococcum, Treboxia, and Apatococcus. This is the algae genus frequency of urban and rural sites. In urban, the highest recorded genus was Apatococcus, and the lowest frequency was Desmococcus. And in rural site, Trentepolia showed the highest frequency and Apatococcus showed the lowest frequency. Apatococcus recorded the highest frequency in urban site. This genus of algae is known for its tolerance to urban environment, including pollution, while Desmococcus exhibited the lowest frequency in urban sites. These genus are more sensitive to pollutants. Here it is stated that only Trentepolia are not present in urban site. This graph in figure 24 shows a slight inclination in the correlation between algae density and ammonia levels in urban environments. As ammonia levels increase, there is a minimal increase in algal density. Algae can utilize ammonia as nitrogen source, which may stimulate growth under moderate concentrations, while in figure 25 depicts a drastic decrease in the correlation between algae density and ammonia levels in rural environments, which means algae in rural areas typically experience lower ammonia levels due to reduced human activities and may be more sensitive to ammonia toxicity, and the higher concentrations could inhibit their growth and survival. Next, on to the conclusion. The ecological effects of ammonia on lichen and algal communities are significant and varied. mainly due to runoff of farms and emissions from industries. Ammonia exposure causes lichen, which indicate environmental health, to grow less, reproduce poorly, and change in community structure. These impacts are both direct, affecting the organisms themselves, and indirect, affecting their habitats. The recommendations are reducing ammonia pollution through farming to a better farming practices, regulations, and new technologies is essential to protect these important communities. Ongoing research and strong conservation efforts are crucial to keep lichen and algal ecosystem healthy and stable. These are the reference for this study. Thank you for your time and interest in my research.